Chapter 3, Topic 3.3, Prokaryotic Internal Cell Structure. The topic objectives for this topic are to describe bacterial cytoplasm and its basic contents, define inclusion and give two examples, describe the formation and function of endospores, describe the structure and function of ribosomes and the cytoskeleton, and compare and contrast the cytoplasm of archaea with that of bacteria. So, we've covered the external structures, now we're going to cover the internal structures. The cytosol, nucleoid, chromosomes, inclusions, endospores, ribosomes, and cytoskeleton. Let's define what cytoplasm is, even though it's not on the list. Cytoplasm is a general term to define everything on the inside of the cell. The textbook describes it as a semi-transparent, fluid, elastic, and aqueous, then turns around and says it includes solid stuff like ribosomes and inclusions. So, for my own sanity, we'll stick to the definition of everything inside the cell, be it solid or solution. On to the cytosol. That is the liquid portion of the cytoplasm, so everything that's not solid. Enough said. Prokaryotes do not have a nucleus, or membrane-bound organelle that encloses the chromosome or chromosomes. Instead, they have a region of cytoplasm where the DNA chromosome is located. Generally speaking, prokaryotes, both bacteria and archaea, have one chromosome and only one copy of the chromosome. Although this chapter doesn't talk about it, I want to introduce you to the concept of plasmids. Plasmids are small circles of DNA outside the chromosome, which makes them extrachromosomal DNA. Plasmids are passed between bacteria through pili. Some bacteria have inclusions, deposits of various chemicals. They may contain reserves of food, such as carbohydrates or lipids, or they may contain cellular waste, such as sulfur. Some photosynthetic aquatic bacteria have inclusions called gas vesicles that store gases in protein compartments. They allow the bacteria to move to the surface of the water during the day and down into the depths at night. Some bacilli are capable of making a structure called an endospore. It is a non-reproductive, resting stage of the bacteria that is able to survive long periods of drying and lack of nutrients. They can survive extremes of temperature, radiation, and most chemical and physical sterilization methods. In a population of endospore-forming cells, a few cells will form endospores even when times are good, but when food resources dwindle, if it starts to dry out, or other poor conditions arise, most of the cells will form endospores to survive the bad times. Basically, a copy of the chromosome is made. One of the copies is walled off by multiple waterproof layers. Meanwhile, the other copy of DNA is broken down. That's why it's non-reproductive. One cell, one endospore. There's no increase in numbers. The endospore is in a state of low to no metabolism. It is in suspended animation. It waits for better conditions. Some endospores have sat on a laboratory shelf for over 50 years and woken up ready to go. Keep all of this in mind when we discuss chemical and physical control methods. You may have learned in Biology 201 that prokaryotes do not have organelles. The reality is that they don't have membrane-bound organelles. For example, they do have ribosomes. Ribosomes are organelles that are composed of proteins and RNA, ribosomal or rRNA, that synthesize proteins within all cells. All prokaryotes, both bacteria and archaea, have ribosomes that have two subunits, a 50S and a smaller 30S subunit. The S stands for Svedberg units, which measures how fast objects sediment within a centrifuge. We don't use grams or other mass units because the objects are too small to put on a scale. When the subunits come together to synthesize proteins, they form a 70S ribosome. No, I didn't make an addition mistake. When the subunits come together, some projections are covered up, and the joined parts move faster than they would if those projections were sticking out. Think of skydivers slowing their descent by spreading out their arms and legs, versus holding their limbs tight to their bodies and diving. Anyway, it goes 50S, 30S, 70S. Eukaryotic ribosomes are different shape and size. They're bigger. They come in 60S and 70S subunits, and they form 80S ribosomes. This becomes important when we talk about antibiotics, so you want to remember this. It will be on the exam too, of course. I also bring up the size and shape of bacterial and eukaryotic ribosomes because the ribosomes of archaea are somewhere in between. Their subunits and complete ribosomes have the same Svedberg units as bacteria, but the shape of the subunits is closer to that of eukaryotes. The amino acid sequence of archaeal ribosomes is closer to that of eukaryotes. This will become more important when we discuss the relatedness of the three domains, bacteria, archaea, and eukaryotes. It used to be thought that bacteria didn't have cytoskeletons. After all, why would they need them when they have a cell wall to provide shape? Well, it turns out that at least some bacilli have cytoskeletons. This micrograph shows some with fluorescent dyes attached to their cytoskeletons. It is thought that the cytoskeleton influences the deposition of peptidoglycan in the cell wall as the cell grows. Some bacteria also use their cytoskeletons to move without flagella. It's amazing what we keep finding in these simple cells. Well, that's it for this topic. I have here a diagram of the average bacterium. It would be good to be able to recognize and be able to label all of the structures with their name and functions, hint, hint. And here is the reminder of what the learning objectives for this topic are.